So yeah, I'm Homère. I'm a PhD student at DTU AQUA in Silkeborg in Denmark, and I'm working on the evolutionary story of the flat oyster, um, both all around Europe, all the population around Europe, but also in the small evolutionary story inside a hatchery or um, a facility that is producing spats, flat oyster spats. Yeah, can you see my screen?
Yes. Yep.
Yep. Okay. Cool. Yeah. So yeah, what I want, I'm really happy to be invited to the NORA Outreach, and the stuff that I'm going to talk about is a case study that I'm working with a high school teacher in Denmark, and the title of that case study is "Rebuild the Flat Oyster Population." I hope you all see the movie Druk or Another Round with Mads Mikkelsen that was last year. Okay, my presentation is full of Mads Mikkelsen in that movie and scenes from that movie. Because yeah, it's a high school teacher from Denmark, so I thought it was pretty suiting. Yeah, so the material in itself, how this project risen from both two. So DTU AQUA, which is a university in Denmark, raised fund from the UN Sustainable Development Goals and that Novo Nordisk fonden to build up new materials, new educational material, which is starting from a PhD student and is a PhD project. And it's a collaborative work with the high school teacher and the PhD student to build up a material. And in my case, because I was not the only PhD student from DTU AQUA that was part of that project, it's about the flat oysters, and the whole thing is to build up material that will fit those sustainable development goals inside. And the end product is for the teacher to use with his high school student already this year. So it's not something that you do and then you archive or not using it. Now it's for using it directly this year. So what is the material that we build up? First of all, the material is an introduction to Australis on some important sustainable goals. I try to fit the whole material into those 4 sustainable goals. And first of all, in that material, I'm introducing how much Australis is important for our culture, with some already in the Stone Age, some fishermen eating a lot of oysters, and we have a record from that. And then the first aquaculture was started with flat oysters, with that Sergius Orata in the Romans era. And still now it's something that we eat, fish, produce, and that is a huge commercial value for many countries, even though now it's more Pacific oysters. The other interesting thing, and so it's only to grab the attention from those high school students that I'm trying to reach large and make them interest risen from that material, and then the other part is that if you want to have a sustainable diet, which is a concern for a lot of people, and I'm sure for also those high school students, there's many definition of what is a sustainable diet, but maybe one common thing about all those definitions is that eating oysters is a good thing to reduce greenhouse gases, but also to have a more sustainable way of eating proteins and many other nutrients as iron, zinc, vitamin B and D. Anyway, we can even call the oyster a superfood. And the last thing of this introduction, and NORA made it really easy, is to show all the ecosystem benefit and also show how much threatened that species is right now. So all these infographics are also present in that material. And so what is the material in itself, and the task in itself that the high school student will have to do? So the task in itself is focus on the Limfjorden population, because it's the last remaining population in Denmark, wild population. And this is Danish high school student material, so I thought it would be suitable that we use the Limfjorden and the flat oysters in the Limfjorden to just build up that material around. And that population is seeing a lot of fluctuation throughout times. This is the last decade and the decade also previously, where you can see that the stocks are increasing and then decreasing. And the same authors that made that graph are stating that the Limfjorden wild population is a small but persistent population. So it's not science fiction to think that maybe this population could be become super depleted. And what if the Limfjorden population is drastically going down for could be many reasons: overfishing, disease outbreak, environmental conditions that are changing extremely. Then what we can do about it? So of course it's an hypothetical case, and I want to restating that because everything about oysters is also highly political, and right now the Limfjorden population is not depleted. But if it was depleted, I think there's two easy choice and easy choice to grasp for those high school students to act on that depleted population. The first option is to import cheap oyster from another country in Europe. The second one is to find ways to maintain local biodiversity, and the underlying question around those two choices is to assess how different those European population of flat oyster are, and actually how genetically different they are, because that's what I'm working on. And my goals with this material is to show the incredible diversity of oysters by including DNA sequences from all those oysters that you see here in that slide. So the Asian flat oyster, but also the pearl oyster, Crassostrea gigas, virginica also. The second goal is to really restating how much Australis is a part of our European identity and history by having a lengthy introduction about it. And the last thing is not the last thing, but another thing is also to make them understand it's also their regional history. And by the way, those two maps are the sampling campaign that we did in my PhD project, and you can see that there is also a strong focus on Scandinavia to sample the most or at least everything that we can sample from Scandinavia and those wild population. And really also is to show how it's done during my PhD project, from the lab work, from the sampling to the lab work, and to the bioinformatic pipeline and tools that I'm using to decipher the evolutionary story of the European flat oyster with a focus on Scandinavia. The goals for Søren, the high school teacher that I'm working with, is mainly to have a material that is suitable for his high school students to work on bioinformatics, because it's something that is not really put in front to work on bioinformatics, and it was his main objective to have something that is usable and that can be done on one session, so not something too huge, and also something that is really illustrated with many perspective. So what we build up together is from my sampling campaign and from my DNA sequencing. I'm doing whole genome sequencing. We decided to have a smaller amount of data in terms of size, and then to reduce that size, we map my whole genome sequencing onto the mitochondrial genome, which is only 16,000 base pair, which is reducing drastically the number of not the number, but the size of all the files, and make them usable usable for online bioinformatic tools, because we don't want to teach all the commands and all the coding and scripting in one session that I'm using for that PhD project, which could be done, but maybe not on only one session, maybe 2 or 3. So we decided to go only with online tools to build up a phylogeny, and that's really what we're doing in that material, with all those DNA sequences, and also the art group. I'm also having a small introduction on NCBI and how to find DNA sequences on an online portal. Then we are having a indication to how building up that phylogeny. And the thing is that by doing that phylogeny is not super sexy, but it's giving clear results, and it's also introducing many knowledge about genetics and how you can separate different species or separate groups within a species. So it's a pipeline to talk about a lot of things, and not in depth, but just like survey a lot of subject in genetics. And the student is, from his first phylogeny to the last one, is going through a trial and error protocol where he can also decide to use certain parameters and try it out, because he got enough time to do it since it's online tools. And he can also go to the raw data that I'm giving him that I give to him to see what is going wrong with it. Like, for example, one of the main thing that the student gotta realize that for some data that I'm giving him, there's some sequences that are not really well sequenced, actually, and you got to go through the raw data and see why that particular Scottish oyster is not clustering well with his other Scottish oysters. And from a phylogenetic tree that is a blob, you can then have a resolved tree by many manipulation and removing sequences that are poorly sequenced. And at the end, you can see yeah, I'm removing a lot of outgroups, and you can see only one outgroup that permits a good interpretation of the relation within the species, and you get a clear result on 4 monophyletic groups, with the Scottish one in purple, the Spanish one in red, um, the Swedish one in brown, and the Nisum green one, which are coming from the Limfjorden. So they get a clear result on the genetic differentiation between those populations by way of monophyletic regional group. And then we can talk after that of restoration program adapted to the site-specific environment, since they are genetically different. So must be some adaptation or signature of selection coming from that environment. That's a stretch, but that's what I want them to understand in a way, even though there's so many translocation and those signatures must be difficult to find, and that's what I'm doing in my PhD project too. But yeah, that's the thing that I want to state to those students. And I think it's also a gateway to talk about a lot of genetic conservation, genetics definition and way of working, which can be the effective size versus the census size, the level of genetic loads. When you talk about hatchery bred oysters, the ancestral genetic diversity, where those who are the direct ancestor of those oyster from the Limfjorden. I'm saying also that it's a young population in the Limfjorden, so where they come from, breeding rate, adaptive potential. There's many ways, and it's also the job of the teacher to choose inside all that toolbox and go with the definition that is more comfortable with to argue with the students. And since the result is not so sexy, and it's a bit dry if it's only something that is a text and online tools, and there is 2 films to introduce the whole material. So 1 film about the lab and the bioinformatic part, and a 2nd film that will come after the material, after the work and the phylogeny work in itself, where it's showing really what we're doing right now in the Limfjorden to support the population. Because of course the 2 choices that they got at the beginning between choosing to import oysters or build up an hatchery and replete the population with local individual, that's an easy choice to understand and to see what is the good answer. But then we got illustration of how it's done, and this is that company from Copenhagen that is doing the 2 films, where they are following me in the lab, and also in the Danish Shellfish Center in Mors, in the north of Denmark, and there's many people that are participating to that movie, and I think it's showing how much how collaborative the work is for those conservation programs. So here we got some spots from the DTU Coastal Group Flat Oyster Hatchery. Those samples are really interesting and important because, like I said, we are also looking at the genetic diversity inside hatchery facilities. Because when you are producing oysters, you want to reduce the impact of inbreeding, and you got to know how many parents are really contributing to the offsprings, and how much inbreeding you are creating by setting some tanks with flat oyster that are reproducing. But yeah, so usually you have more inbred individual in a flat oyster hatchery compared to the wild flat oyster. And finally, yeah, I just want to state that it's not a finished product, so it was not tried on the high school student. The two movies that I'm talking about are not done yet, and the whole links between the parts of that material are not connected yet. So it's a work in progress, and yeah, that's the last part, which is not the movie. That's myself filming, so that's why it's so bad, shaky camera. But yeah, that what I like about the material is that you see a lot of part of the of a comprehensive restoration work, or scientific the scientific part of a restoration work. We don't see the restocking campaign or anything like that, but I think we can follow a thread on how genetic information is found and then given uh, to hatchery, and at the end can be used or not for restoration program. And yeah, I want to thank all those people here that are a part and help a lot in that material. So my supervisors, Dorte Begvold, Jacob Emmeronsen, Camille Sorel, that help a lot on the part of Morse and the hatchery, and also the scenes that we shoot at the hatchery. Thanks also to Philin for reviewing the material at the preliminary steps of that material. Soren, the high school teacher, that of course that is the main part of that material, and then there is Christian Risager that secured the fund and is the instigator of the whole funding for those materials. Pernil, who did all the movie and shooting. Pascal Barraud, that is a super great actor, but also the master technician of those flat oyster in Morse. And Jens, the director of the Danish Shellfish Center, who gave an interview to us and the movie team. And that's the ref. Thanks a lot.